Hello, I'm Ayushi Tiwari from Sachdeva Global School, and I will be explaining you about biotechnology. So without any further delays, let's get started. Before going into the depth of the topic, let's first understand what exactly is biotechnology. If you want to define it in a layman's language, then it is a technology based on biology. But biology doesn't accept such definitions. Therefore, it is the science of using living organisms or products of living organisms for human benefit or the benefit of his surroundings. Now, since we know what is it, we must also know what comes under biotech. So it's very simple. Things that we see around us or maybe ever heard in our lives comes under biotech. I just mean from making bread, curd or wine to developing DNA vaccines or correcting defective gene, all of this comes under biotechnology. There are always one or the other techniques to accomplish something. It is for biotech too. It also has two core techniques on the basis of which it works. One of them is genetic engineering and the other one is bioprocess engineering. Genetic engineering, as we know it, is the direct manipulation of the genetic material to alter the phenotype or the characteristics of the organisms. Bioprocess engineering, on the other hand, is the maintenance of sterile environment. Okay, so sterile here means contamination free or an environment which is free from bacteria or the microbes. For the chemical engineering processes which are required for the growth of desired microbes in large quantities to get the products required. The products could be antibiotics, vaccines or enzymes. Now let's understand genetic engineering a little deeper. There are various techniques to perform genetic engineering. Recombinant DNA, about which we will be understanding in just a bit. Gene cloning, which is making an exact copy without making any changes to the genome. And gene transfer, which is the insertion of unrelated genetic information in the form of DNA into cells, comes under this. For now, let's come to DNA. Stanley Cohen and Herbert Boyer accomplished the development of the recombinant DNA in 1972 through isolating the antibiotic resistance gene by cutting the DNA at specific locations. The cutting of the DNA is only possible with the restriction enzymes. Restriction enzymes are a major part of recombinant DNA. The role of restriction enzymes, in short, is to cut the DNA at specific locations. Restriction endonuclease is one of the restriction enzymes. HIND2 is the first restriction endonuclease. After certain experiments and research, it was found that HIND2 always cut DNA molecules at specific point with the sequence of 6 base pairs. The specific base sequence at which the DNA gets cut is what we call as the recognition sequence. Restriction enzymes belong to a larger class of enzymes called nucleases. Nucleases are of two types, exonucleases and endonucleases. Exonucleases removes the nucleotides from the ends of the DNA. And endonucleases makes cut at specific locations within the DNA. Each restriction endonuclease recognizes a specific palindromic nucleotide sequences in the DNA. Palindrome is a word used for such kind of words which remains the same no matter you read it from left to right or right to left. For example, level or race car. From our previous knowledge, we know that DNA is negatively charged, so the fragments of DNA can be separated out by using gel electrophoresis. In this technique, an electric field is provided due to which the negatively charged DNA fragments moves towards the anode. It's time to bring in the cloning vectors. These vectors at present are developed in such a way that they enable easy linking to foreign DNA and also helps in the selection of recombinants and non-recombinants. Now we will understand about the features of cloning vectors. They are: first, origin of replication, which is the site from where the replication of DNA gets initiated. Second is selectable marker, which helps in identifying and eliminating non-terminants and simultaneously permitting the growth of transformants. Third is cloning sites. These are just the recognition sites where the cloning occurs. Now, recombinant DNA is not done yet. We are still left to understand the processes involved in recombinant DNA technology. So the first step is to isolate the genetic material. This is achieved by treating the bacterial cells or plant and animal tissues with certain enzymes. Next step is to cut the DNA at specific locations. This, as we discussed just now, is done by restriction enzymes and the fragments will be separated by gel electrophoresis. Third step is the amplification of the gene of interest using PCR. Here PCR stands for polymerase chain reaction. In this reaction, multiple copies of gene of interest are synthesized using two sets of primers and the well-known enzyme DNA polymerase. Fourth step is insertion of recombinant DNA into the host cell or organism. This can be done by the selectable markers. Fifth step is to obtain the foreign gene product. This can be obtained by using the bioreactors. Bioreactors are just vessels in which raw materials are converted into required products biologically. And last but not the least step is downstream processing. This is a series of processing in which the product obtained is separated and purified. The future of biotechnology is strong. We envision a day when breakthrough drugs lead to a world without cancer or AIDS, a world where there is sustainable development that will tackle energy, food, and environmental needs without compromising the Earth's resources or its future.